Hello, everyone. I'm Dr. Linda Hisatsri, an assistant professor of laboratory medicine and pathology, um, and the director of the Molecular Technologies Lab in the Division of Laboratory Genetics and Genomics at Mayo Clinic. I'd like to welcome you all today to our Future Direction of Lab Medicine webinar on pharmacogenomics. Why is everyone talking about DPYD? It is my sincere pleasure to introduce our speaker today, an internationally recognized expert in the pharmacogenomics space, Dr. Anne Moyer. Dr. Moyer is a professor of laboratory medicine and pathology, as well as an assistant professor of pharmacology, and is a molecular pathologist in the Division of Laboratory Genetics and Genomics at Mayo Clinic as well. And she also co-directs the Molecular Technologies Laboratory. And with that, I will hand things over to Dr. Moyer.
Well, thank you for that great introduction. And then also thank you to everyone who has joined today. I'm really excited to talk about one of my favorite topics, pharmacogenomics. And specifically, we're going to be focusing a lot of the time on one of the genes that's really hot in this space right now, DPYD. And hopefully by the end, you'll know why everyone's talking about DPYD. I have no disclosures. And so for our learning objectives, we're going to hear a little bit about pharmacogenomics as a whole, explain why DPYD testing is important in oncology, and we'll debate some of the pros and cons of sequencing-based pharmacogenomics because I think that's where the field is headed. So I really wanted to start out with a couple of patient stories to make sure that everybody understands how important this type of testing is, and especially DPYD. So what you see here is an article that was published a couple of years ago about a family that's in Canada. And the man in the picture is actually a physician in Canada who had developed colorectal cancer. And ultimately, instead of being treated by a fluoropyrimidine medication to help cure his cancer, he ultimately died as a result of the therapy. His whole family is made up of physicians, and they actually didn't know about DPYD testing being an option that could have potentially helped predict this toxic reaction and may have helped them recognize that he might have benefited from a dose adjustment. So I think this is a story to keep in mind, and we'll mention this family again later in the talk. In addition, here's another example. This woman is Carol, and she actually had developed metastatic breast cancer, and kind of the same story. She ended up taking a fluoropyrimidine medication, the goal of which was to help treat her cancer. But instead, you can see in this story, they describe some of the very specific complications that she experienced. So she ended up with having severe diarrhea and nausea, and probably one of the more significant adverse reactions being these horrible sores. We call it mucositis in her mouth that prevented her from being able to eat and drink and ultimately speak. And so this was just an absolutely miserable way to have this type of reaction occur. And again, this is a situation where this drug was intended to help treat her cancer, and instead she ended up developing toxicity from it that was quite severe and ultimately was fatal. She ended up passing away from the therapy rather than the cancer. And so both of these stories are patients who took fluoropyrimidine medications. And we're going to talk a little bit more about DPYD because DPYD testing is something that can be performed upfront before the patient gets one of these medications. And in some cases, we may be able to predict this severe adverse reaction before the patient experiences it. And in that case, give them a dose reduction, but more to come on that. But I just really wanted to mention these couple of patients because part of why everybody is talking about DPYD today is because of patients like this, and in particular, their family members. There are a lot of patient advocacy groups that have been really spreading the word about DPYD and why it's so important, and we're going to touch more on that as we go. So if you think about just adults in general in the United States and throughout the world, it's at least 60% of adults who are taking medication daily, and probably everybody will at some point in their lifetime. And adverse drug reactions are actually somewhat common. In 2022, it was well over 1.25 million severe adverse drug reactions that were reported and nearly 175,000 deaths. So, if there's something that we could do to help understand which patients are more likely to experience one of these reactions, that would be really helpful for patients so that the medications that they're taking to actually help them don't end up harming them. And so that's what pharmacogenomics is really focused on. So, how could we reduce the incidence of adverse drug reactions? Well, one thing you want to do is start thinking about pharmacology as a whole and think about what are all of these variables that impact medication response. Several of them are shown on the screen. There are many, many different variables, but among them, one of them is genetics. And one thing to keep in mind is with pharmacogenomic testing, it's really exciting. It's a tool that can help us predict some of these adverse reactions. But again, it is not the only variable. So we just have to keep in mind when we do pharmacogenomic testing, we can't forget about everything else that's important in pharmacology. So, what's the goal of pharmacogenomics? As a whole, our goal is to use genetic testing to select the right dose for the right patient at the right time. And so, basically, that's what we're trying to do with pharmacogenomics. And this field has been around for quite some time. And so, I wanted to show you a little bit of a timeline because I think sometimes people hear about pharmacogenomics and they think it's brand new. But indeed, the first publications were from the early 1960s in this field. And then by the late 1990s, the bar graph here is showing the number of publications in PubMed. You can see that we're starting to see a little bit of an increase in articles published about pharmacogenomics, and this corresponds to when the early adopters began clinical testing. Usually it was one gene at a time at that point, and not used very widely. By the early 2000s, the Human Genome Project was completed, and that's when you're starting to see really a big uptake in the number of publications in the pharmacogenomics space. And by the 2010 through 2020s, this is when multiplexing really started to increase, as well as clinical adoption of pharmacogenomic testing, and probably even more importantly, standardization efforts started to come into play. And this is really important because if different laboratories were doing pharmacogenomic testing, everybody was doing things their own way. And so it was really hard, both from a clinical perspective and from a research perspective, to compare results from one lab to another. And so these have been really helpful for the field. And so at this point in time, we really have a strong foundation of scientific knowledge and evidence for clinical testing of pharmacogenomics. And so it's really starting to take off, and we're seeing a lot more test orders come in clinically, especially for genes like DPYD. So, where are we going in the future? Well, I think we're going to see more and more integration of pharmacogenomics into standard molecular laboratory testing. It's really going to hopefully become mainstream and something that's commonly used for patients. And what I'm really excited about is over time to see a transition from genotyping based approaches to sequencing. And we'll get to that at the very end of this talk. So, one thing that's really important to know about with pharmacogenomics is that most patients will have at least one potentially actionable pharmacogenomic variant. So, at Mayo Clinic, we performed the Right 10K study back in about 2000 or 2022. And in this case, we sequenced over 10,000 Mayo Clinic patients for 12 pharmacogenes listed here. And in this pie chart, what you can see is the number of genes that each of these patients would have had a variant in. And so, of our 10,077 patients, only 0.6% of these individuals did not have a single genetic variant in any of these pharmacogenes that we felt would be potentially clinically significant. But there were other patients that had variants in one gene, about 15, 16% in two genes. Most patients had variants in three or four genes. And the most we saw was there were a few people that had a variant in eight of the genes. But at any rate, the thing that's interesting about this is each of these genes can either metabolize or be involved in the mechanism of action for different medications. And so some of it really depends on luck. So if you happen to only have a variant that's potentially clinically actionable in one of these genes, and you never end up taking a medication that happens to be metabolized through that pathway, it may not be clinically significant. However, sometimes it's just as important to know that you're a normal metabolizer and should theoretically be able to take a normal dose as it is to know that you would need a reduced dose. But again, remember that there are always other variables at play as well. And so I think the take home message here is that pharmacogenomic testing could be important for pretty much all patients. So, what exactly is this pharmacogenomics? Again, we're using genetic information to guide medication selection and dosing. And most of the time, we're talking about drug metabolizing enzymes, but that's not all that pharmacogenomics is limited to, but we'll talk about them first. So, every time a patient takes a medication, you can see the drug concentration on the y axis would increase with each dose, and the patient will metabolize away some of that medication between doses. And the goal is you really want their concentration of drug to stay within the therapeutic window because that's where the drug should be effective for them. And hopefully, they will be avoiding any medication dose related toxicities. And unfortunately, some patients, though, will actually metabolize that same medication, same dose, a little bit less quickly. So over time, they may see an increase in the concentration if they're just given a standard dose and may end up with levels that could be related to these concentration related toxicities. And in other cases, some patients might metabolize the medication a little quicker than average. And in that case, you're worried about maybe they won't have enough of the medication in their system and may experience lack of efficacy. So, how we end up describing this in clinical terms in pharmacogenomic testing is using this paradigm of the metabolizer status. So, the patients who metabolize medications as expected, we call them normal metabolizers. The people who have very little drug metabolism capability with that particular enzyme are called poor metabolizers, and people who may be able to metabolize the medication faster than average are called ultra rapid metabolizers, with rapid being between normal and ultra rapid, and intermediate being between poor and normal. So, if you're thinking about this in terms of genetics, for the most part, patients who are poor metabolizers will usually either be homozygous or compound heterozygous for deleterious variants. Patients who are intermediate metabolizers might be heterozygous, or they might have genetic variants that just decrease enzyme activity to a lesser degree and could still be homozygous or compound heterozygous in that case. And then the rapid and ultra rapid metabolizers either have variants that end up impacting the transcription and then ultimately translation of the protein, or they might have gene duplications. But it's not just drug metabolizing enzymes. There are also genetic variants in drug targets, and there are also specific HLA molecules that can predict idiosyncratic drug reactions. And so, really, with pharmacogenomics, we're wanting to use the genetic information to predict which group the patient is going to belong to, and that's going to help us guide drug selection and dosing. So, if we're thinking about pharmacogenomics, pharmacokinetics, that's what does the body do to the drug, that's a lot of what we're doing at this point. So, the absorption, distribution, metabolism in particular, and excretion of medications. And just a few examples would be CYP2C19 is a gene that encodes an enzyme that metabolizes clopidogrel, DPYD that you're going to hear more about for fluoropyrimidines, and TPMT and NUDT15 for thiopurines. But there are many more than this. Pharmacodynamics, in contrast, is what the drug does to the body. And so these are going to be the proteins and molecules involved in the mechanism of action of the drug. Probably the most famous example being VCORC1 for warfarin. And then, as I mentioned, these HLA molecules. And so they are associated with very specific drug reactions that are very much typically severe skin reactions. So DPYD is going to come up a bit more today. So, what does this look like when you're thinking about a clinical laboratory? Well, when we do our test, we first identify the raw data. So, that's how we identify individual genetic variants that the patient has. And then we need to convert those into haplotypes and diplotypes, or you'll see them on the report as star alleles. So, that's going to be like star one, star three. All that's doing is that's nomenclature to help us keep track of what variants were detected. And this is standardized. And then we use this to convert into the predicted phenotype and activity score. So that's where intermediate metabolizer or poor metabolizer comes in. And then ultimately, we'll convert that into medication recommendations, such as maybe decreasing a dose or considering an alternate medication. And I just wanted to touch very briefly on activity scores. These are really just a more granular way to express this whole spectrum from poor metabolizer through ultra rapid metabolizer with a numeric score. And so if you see those on a report, then you may be able to line that back up with either specific medication recommendations from some of the professional societies or looking on the report that can help you figure out what dosing you might need for your patient. So it's just a more granular way to express this same concept of the predicted phenotype. So let's talk more about fluoropyrimidines. So these are medications used for the treatment of colorectal cancer and other solid tumors, just like you saw in those couple of patient stories. And unfortunately, patients can experience these severe adverse reactions. And it's thought that it's about 20 to 30% of patients may experience a severe reaction. Usually it's thought to be less than 1% that are fatal, but even these severe reactions, they're just miserable for the patients. And so if there's a way we could prevent these, whether they're severe or fatal, we would definitely want to do that. And as you saw in the study, or in the patient example, myelosuppression, diarrhea, nausea, vomiting, mucositis, and even cardiotoxicity are among the adverse reactions. So, how could we predict these? Well, you're going to see in a minute that the enzyme dihydropyrimidine is dehydrogenase is an important enzyme in metabolism of fluoropyrimidines, and this is encoded by the DPYD gene. And so patients that have partial or complete deficiency of DPD end up being at increased risk. And so it's thought to be about 2 to 8% of individuals have a partial deficiency. In many cases, these might be heterozygotes, and about 0.2% have a complete deficiency, leading to potentially severe toxicity. This would include the compound heterozygotes and homozygotes. Now, it's not exactly there's not a perfect genotype phenotype correlation, and that sometimes the heterozygotes can have more severe toxicity than you might expect based on what you would think their enzyme activity may be. And so this is just one thing that's really important to be aware is if you look at these numbers, 2 to 8%, another 0.2%, and then up here we were talking about, well, 20 to 30% of patients can have these adverse reactions. Well, we can do a really good job with genetic testing where when we find a variant, it is highly predictive of a patient potentially needing a dose reduction so they don't experience that toxicity. But we unfortunately cannot identify all of the patients who will go on to have a severe adverse reaction. So I think that's important to be aware of. This testing is very beneficial for the people when we do find a variant, but you still have to watch the patient carefully. And I think we still need to do some more research in this area so that we can identify more of these patients. So, what are these fluoropyrimidine medications? They've actually been around for a really long time. These are anti-metabolites that are used in cancer treatment, and they can interfere with DNA production and therefore cell division and tumor growth. And if you just look at the structures of them, here's the 5-fluorouracil, and then capecitabine is similar, except it has some protective groups so that it can be administered orally. Well, it looks really similar to our pyrimidines, thymidine and uracil. And so basically that's kind of how these drugs are working is they're being incorporated in place of these compounds. So the 5-fluorouracil or the fluoropyrimidines in general, they are detoxified by DPD, and this happens generally in the liver. And so it's about 80 to 85% of the administered dose will be converted by DPD to inactive metabolites. And then the rest of the dose, about maybe 20-ish%, will go into the cell. And then at that point, that's how it can have its mechanism of action being incorporated into DNA and RNA, ultimately leading to cell death, as well as disrupting the trinucleotide phosphate pools. So a DPD deficiency, basically, if you are not able to send as much of the medication through the liver to be detoxified, that means more of it is going to go into the cell and have these effects, which again, you do want to kill the tumor cells. That's where hopefully this activity is happening, but you'll have enough of this fluoropyrimidine floating around to be able to enter other cells as well. And so if too much of it goes down this pathway, that's where your toxicities are coming from. And so you can actually adjust the dose for people who are known to have decreased or no DPD activity. So if you just give them less of it, since you can't shunt it down this pathway, that means there will be less available to go into the healthy cells as well as cancer cells. So, thinking about the genetics of DPD, so DPD is encoded by the gene DPYD, and it's on chromosome 1. It's actually a quite large gene, and we know that genetic variation in DPYD is a main contributor to variability in DPD activity. There are probably other variables as well, but this is definitely one that's important and something we can test for today. So, we can perform genetic testing, either genotyping or sequencing, to identify variants that predict the risk of fluoropyrimidine toxicity. So, one thing to note, though, that's kind of interesting and not something that we think about with all of our pharmacogenes, but there's also an inherited disorder called DPD deficiency. And this is thought to be very rare, and it has a very wide range of severity, with many patients having absolutely no phenotype at all. It's thought to be autosomal recessive, so presumably both copies of someone's DPYD gene would be impacted. And when it is symptomatic, it tends to manifest very early in infancy with severe neurologic phenotype, including epilepsy and delayed development, hypertonia, and multiple other features. But what's really interesting is this is a disorder that we consider to be a low penetrance disorder. So what I mean by that is that most people who have deleterious DPYD variants will not develop this disease. They will be walking around perfectly fine in life until they end up developing a cancer if they do. And in that case, these people are people that were otherwise healthy. And then when you treat them with a fluoropyrimidine medication, they can still develop this severe toxicity, even though they absolutely didn't develop this other phenotype. So I just wanted to mention this just so that everybody is aware that this is out there. So we do testing in addition to for pharmacogenomics. We can also do diagnostic testing for this condition. But again, this is a condition where I think more research is needed to better understand why so many people can have genetic variants and not actually manifest the disorder. So, a little bit of a brief timeline thinking about fluoropyrimidines and DPYD. So, as I mentioned before, these drugs have been around for a long time. It was back in the 1960s that fluoropyrimidine first received FDA approval. And then it wasn't until about the 1980s that 5-fluorouracil toxicity was linked to low DPD activity. And by the mid 1990s, the molecular mechanism was starting to be understood. And a few years went by, and there's a CPIC guideline that I'm going to show you in a minute that came out in about 2017. And in 2022 and 2024, the FDA labels were updated to indicate that patients that have an absence or near complete absence of DPD are at increased risk of these adverse drug reactions. And then in the spring of 2025, the FDA issued a safety announcement to really increase awareness of these label updates. And so this has really started getting everybody talking, but we're going to come back to what happens after that because this has been a really active area the last few months. So here's the Clinical Pharmacogenetics Implementation Consortium guideline that I mentioned. And this is actually an update one in 2017. So there had been one before this. And basically, the CPIC, it's a group that works at putting together guidelines and information on pharmacogenomics so that it can be more broadly accessible and so that this can be better implemented. So they've really been leading a lot of those standardization efforts that I mentioned. And their goal is so that if somebody has genetic information, they know how to use that when they're thinking about pharmacogenetics. So what you can see here is just a screenshot from their guideline, and you can see the phenotype that I mentioned before, which will be on your pharmacogenomic testing report. So normal metabolizer, intermediate metabolizer, or poor metabolizer. And based on what that result is, there's a dosing recommendation. So for the intermediate metabolizers, you can see that they would recommend reducing the starting dose based on the activity score. And that's what I had mentioned as another more granular way of describing the phenotype. So potentially a dose reduction of somewhere between 25 and 50%. And then with the poor metabolizers, again, there are recommendations to potentially even consider avoiding the use of fluorouracil if there's something else that may be appropriate for that patient. And so I think this has really helped people to be able to use the genetic testing results. So why wouldn't we test patients for DPYD? So could the testing delay therapy? Could it be too expensive and not covered by insurance? That's definitely always a concern. What if reducing the patient's fluoropyrimidine dose leads to ineffective treatment? I mean, we're using these medications to try to treat someone's cancer, so we definitely would want to make sure that we're not going to underdose them, and that's been something that's been very much a concern in the field for a number of years. And there are guidelines that are commonly used by oncologists, and recent or until recently, they had not been encouraging testing. And I think the main concern was exactly this one, worries about what if we end up undertreating our patients. So there have been some new developments, though. In 2022, there's a meta-analysis that I just wanted to highlight where they showed that pharmacogenomic guided treatment for fluoropyrimidines reduced the incidence of grade 3, 4 overall toxicity. It reduced the diarrhea, and there was no statistical difference for the overall response rate, nor the difference in stable disease. And so I think these were the pieces of information that people were really interested in seeing because people had known for a while, well, if you do reduce the dose, the toxicity is less, or the patients don't experience toxicity if they had a variant. But we really wanted to make sure that we weren't going to be under treating them. And other studies have come out since then that have supported this as well. And then this type of work, these newer studies that were demonstrating no change in outcomes for the patients who had a pharmacogenomic guided dose reduction, ended up resulting in the NCCN group revising their guidelines. And so at this point in early 2025, they suggested that testing for DPYD genetic variants should be considered prior to fluoropyrimidine therapy. And after discussions regarding risk assessment, patients could choose if they wanted to get DPYD genetic testing, but there was no specific test recommended. And they also indicated that there is insufficient data to inform dose adjustments for some of those DPYD variants that are detected. And so we'll talk more about that later. So this is probably referring especially to things like variants of uncertain significance. So this is a really big step forward that the guidelines are mentioning that it's probably worth thinking about doing this genetic testing or at least talking about it with patients. So I extended out the timeline. So now in the fall, or actually, sorry, in the spring, the NCCN guidelines were updated. And then ultimately in October, just recently, the FDA updated the FDA label for capecitabine with a boxed warning and recommending DPYD testing. So what does that look like? Well, here's the capecitabine FDA label. And remember, capecitabine is the version that can be taken orally. And so here's our boxed warning that now exists. And I guess it always existed before about some of the coagulation parameters. But what you can see that's included now is that they mention that there's a severe or serious adverse reactions or death may occur in patients with complete DPD deficiency. Test patients for genetic variants of DPYD prior to initiating Xylota unless immediate treatment is necessary. And you should also be avoiding it in homozygous or compound heterozygous patients that could be resulting in complete deficiency. So one thing to be aware of here is they're really focused on people with complete deficiencies. So that would be the poor metabolizers versus the CPIC label also did have some guidance for patients who would be intermediate metabolizers. So that's one difference just for awareness. But what the big change is here is now instead of just mentioning that genetic testing was available, they're really recommending that you should be doing this testing up front unless there's a need to immediately treat the patient. Now, the 5-fluorouracil label was updated in 2024, and it has not been updated to include this boxed warning yet, but it has the slightly softer language that still talks about DPYD variants and their importance, but doesn't necessarily actually recommend testing at this point in time. It just ends up mentioning that this is a possibility. So, why is everybody talking about DPYD now? So, newer studies demonstrate that efficacy is similar when the dose is reduced based on pharmacogenomics. It makes a lot of sense. Patients with decreased DPD activity will clear less of the drug, so the actual drug concentration should be pretty similar in patients who have a pharmacogenomic variant and end up having a dose reduction as it is to patients who did not require a dose reduction because they had no genetic variant. These tests are widely available. The results can be returned quite quickly, usually within about a week, and the insurance coverage is really increasing, and this includes Medicare coverage at this point in time. So it's a really exciting time to see that it's being recommended and available for patients. So the other thing that's really important to be aware of is the patient advocacy groups. I don't think that a lot of this would have happened without them going out there and being vulnerable and sharing their stories about their loved ones that were impacted negatively by fluoropyrimidines and trying to figure out, well, what can we do differently that might help prevent this for some patients? So I think that's something really important just to know that all of their work is definitely being heard. So at the end of the day, no one wants their patient to suffer or die from the medication that's being used to treat their cancer. So I think everybody really wants the same thing. And so it's nice to be able to see all of this progress in this area. So now we're going to talk a little bit about designing a pharmacogenomic test. So this is really important because we're still doing genotyping for pharmacogenomics. So the gene content and alleles that are included end up being really important. So variant selection. If you're genotyping, you have to design your assay to include the individual variants that you're going to need to detect. And you have to validate that each variant can be detected. You have to build each one into your workflow. And this is, again, really important because sometimes the allele frequency can vary by ancestral populations. So in some populations, some variants might be more common or less common. So just an example for 3 DPYD variants that are relatively common, you can see different populations around the world listed in the columns, and you can see that some of these variants tend to be more common in some populations than others. And so if we aren't really cognizant of this and thinking about it while we're designing our test, we could accidentally introduce healthcare disparities by not being inclusive of the alleles that are common around the world. And so this is definitely a limitation of genotyping, but it can be overcome with careful test design. So when laboratories are using different test designs, they might end up seeing different results. So on the top, I've got laboratory 1 and on the bottom, laboratory 2. They're both doing great work. Their tests are doing exactly what they're supposed to do, but their test design is different. So the gene DPYD is the blue line and each orange arrow is where a variant was assay was designed. And so you can see if the variant is where this yellow mark is. In both cases, the two labs, they're going to result out the exact same thing. But the issue is that if the variant happens to be located in a different position within the gene, like you can see here, laboratory one won't detect this variant. They'll consider this patient a normal metabolizer, but laboratory two, they will detect the variant, and this would result in them recommending a dose reduction. And so in this case, both tests are performing exactly as expected. They're producing accurate results. Both labs are doing a great job, but different genotypes are reported at the end of the day. Different phenotypes are reported, and this results into different therapeutic drug recommendations. And so this is something that's really important because as a field, we need to be more consistent so that we're making sure that we have tests that are going to be similar across laboratories so patients don't get very different results from different labs. And so this is where the Association for Molecular Pathology Pharmacogenomics Working Group has come in. Their goal was to develop recommendations for a minimum set of variants or a must-test list that would be included in clinical genotyping tests to promote standardization. And so these are used by the general pharmacogenomics testing community so that we can design our tests more appropriately. But interestingly, they're also being used by the stakeholders who use pharmacogenomic testing at this point to make sure that the test includes what it should include. So who's in this group? Well, there's subject matter experts that are representing the pharmacogenomic testing community throughout the US and Europe at this point, as well as representation from multiple different professional societies. So it's been a very collaborative effort. So, what are these recommendations? Tier one, those are the must test alleles. They should be well characterized variants. They have an appreciable minor allele frequency in any patient population. Keeping in mind that in the US, our population is quite admixed, there have to be available reference materials, and it has to be technically feasible for a laboratory to be able to detect the variant. And then tier two is really an extended panel, so they're meeting some of these criteria, but not all of the criteria. So these are the nice to have variants that we would like to see included if we can. And so the group started out with CYP2C19 and expanded to CYP2C9, and then kept going from there, and ultimately recently published guidelines for DPYD. And so this was published in October of 2024. And so laboratories out there around the world are working on updating their existing DPYD tests, and labs that are implementing this testing are looking very closely at these recommendations as well. And so here you can see the tier one variants and then the tier two variants, as well as their population frequency. So again, the tier one variants tend to, for the most part, be a little bit more common in the population, as well as meeting those other criteria. So, some limitations of DPYD testing. Well, remember, as I mentioned before, this isn't the only variable that contributes to toxicity, so we need to keep that in mind. But also, there's some patient-specific variables. So, if the patient recently had a blood transfusion, you want to pay attention to whether it was leukocyte reduced. Most of them are in the US and therefore probably won't impact genetic testing. But if there are a lot of white blood cells present, that could be an issue. Allogeneic hematopoietic stem cell transplants. Well, this is pretty important because, again, DPD is expressed in the liver. And if you're testing someone's whole blood and they had a stem cell transplant, you're not really testing the DPYD that is reflective of what would be in their liver. And clearly, then, if they had a liver transplant, same sort of scenario here. So if you run into any of these issues, it's worth reaching out to the lab and we can walk you through what the most appropriate next steps would be in those cases. The other thing that's really important to be aware of, remember, we're using DPYD in the setting of cancer, and therefore the tumor tissue might be getting sent in for other guided therapies. And in that case, if the tumor is being tested for other applications, you might think, well, this is supposed to be germline genetic variation. Maybe we can just test the tumor for DPYD and get it done all at the same time and not have to send in a blood sample. We really don't recommend that because, again, most of the metabolism happens in the liver. Maybe there's something going on in the tumor, but not significant compared to the liver. And the tumor could potentially have somatic variants that could result in something that would cause you to want to do a dose reduction that wasn't appropriate, or they could lose a variant allele along the way, resulting in a false negative. So really avoid testing tumor tissue in this case and go for a germline source like blood. So current pharmacogenomic testing, thinking about DPYD and beyond, There's been, as I mentioned, a lot of historic variation among laboratories, but it's very much been decreased due to the field working together so hard on standardization efforts. So PharmVar is a group that has created the star allele definitions, or curates them rather. The Association for Molecular Pathology and Partners, those recommendations for allele selection, CPIC that I mentioned earlier with the standard nomenclature, as well as a lot of guidelines on how to use these results and the standardization and genotype to phenotype translations as well. And then the regulatory agencies, in addition to CPIC and the Dutch Pharmacogenomic Working Group, are all putting out dosing and drug selection guidance. So, what's not standardized today? Well, if you're ordering a panel-based test, the genes that are included in the panel are not standardized. You can see a lot of variety there still. And one thing just to be aware of when you're doing pharmacogenomic testing, it's a little different than inherited genetic testing. So it's got some nuances, and it's important to have the laboratory have expertise in pharmacogenomics. So here's just an example of what a report might look like for the genotyping test. And in this case, you can see the patient has a phenotype of intermediate metabolizer. Their activity score is 1, and then there's detail about the specific variant detected. And then generally, labs will include some description of what exactly that means for the patient, as well as methods and disclaimers. So you would want to be able to go and match this back up with the CPIC guideline. So you're looking and you're like, okay, intermediate metabolizer. So this is the section of the guideline we're looking at here. Activity score of 1. So they're suggesting you might want to reduce the dose by about 50%. But again, remember, we're just looking at genetics here, and all of the other variables remain important. So back to this family that we talked about before, and Dr. Kapoor here. So one of the things that I thought would be important to mention is that even if all of the laboratories adopted all of the AMP Tier 1 and Tier 2 DPYD variants, the variant that this family happens to have still would not be detected. That's kind of important to be aware of because, I mean, we've got all of these patient advocacy groups that really want something to be done so that we can avoid these horrible toxicities that their family members have experienced. But in this case, we still wouldn't detect it if we had done the AMP Tier 1 and Tier 2. So, what could we do instead? Well, this is where sequencing-based pharmacogenomics, I think, is really important. This is where we can detect the variants that are not detected by genotyping. So you can't clearly design a genotyping test to detect every single possible variant out there. It's just not feasible. That's where it's important to use sequencing-based tests instead, because they can detect common variants and rare variants. So here's an example where if the variant happens to be here, and again, our genotyping assay is looking where these specific orange arrows are, this is just not going to be detected at all. And this patient is going to be getting a report that's going to say, yep, you're a normal metabolizer. Versus if we do a sequencing assay, because we're looking at the entire region of interest, we can identify that variant. But the biggest con to that is once you've identified that rare variant, sometimes these are really difficult to interpret. There might not be a lot of clinical information out there, or sometimes no clinical information out there. And so it's hard to know exactly what to do with that variant. Does it decrease enzyme activity? Does it not decrease enzyme activity? We don't really know if it hasn't been studied, and especially if it's a variant that's unique to that particular patient and hasn't been seen before. So that's, I'd say, the biggest challenge of sequencing. So, some other advantages to thinking about sequencing in the pharmacogenomics space. Again, we really want pharmacogenomics to be mainstream because we think it's really impactful and it could really help a lot of patients. But most of the diagnostic testing laboratories that are out there today are doing a lot of sequencing based testing and not quite as much genotyping anymore as historically. But again, for pharmacogenomics, we're mostly still genotyping. So maybe we could make it so that more labs could get into the pharmacogenomics space if we harmonized pharmacogenomics with a lot of those other diagnostic testing workflows. We would potentially have fewer instruments in the laboratory if we were just able to use sequencing based approaches. And so the test design would be a bit simpler. We wouldn't have to sit there and think about, well, did we include all of the variants that we really need to so that we're hitting on what's common in patients around the world? And we would also not have to worry about missing those rare variants in this case, too. But keep in mind, if we're going to do in general for pharmacogenomics, not DPYD specifically, because it's a pretty well behaved gene, but in pharmacogenomics, we've got some genes that are a bit more challenging. They're homologous, they have another gene that's very similar to them that can complicate testing. testing. And so we have to be careful of that if we're going to use sequencing-based approaches. And we also have to be really careful because a lot of times in diagnostic testing, common variants are probably not going to be causing rare disease. But in pharmacogenomics, those common variants can be really clinically significant. So as a laboratory, you have to be careful to not filter those out. And the other thing that's important is there are some well-established non-coding variants in pharmacogenomics today. And so, if you're using exome-based testing, you might be missing some of those. But in the future, hopefully, we'll be also including and understanding more of the non-coding regions of these genes as well. And so, I think that's going to be a little bit harder to get to the non-coding variants because that's still a little bit of a challenge even for diagnostic testing. But just keep in mind that even if we're switching to sequencing, we may or may not be including the introns in some of these genes at this point. So reporting is going to definitely take more thought as labs switch to sequencing, though, because they've got multiple different strategies that they could consider. Do they just want to report the variants that are listed in PharmVar and CPIC? Do they want to include variants that are clearly going to be impacting the function of that protein? Or do they want to include all of the variants? And then also thinking about copy number variation, so whole genes or parts of genes being deleted and duplicated. Right now, a lot of that's not in scope for genotyping for pharmacogenomics, with a few exceptions. But if we switch to sequencing, we may be able to detect these across more genes. So going back to this Mayo Right 10K study, in this case, what we actually did is we decided to sequence these patients. So it was 10,000 patients. And again, we were clinically reported on 12 genes. There was a special algorithm for one of our challenging genes, CYP2D6, and the rare variants were actually flagged and then classified and interpreted. So we didn't ignore them. So it's truly a sequencing-based test. So, a follow up study that we performed was looking at comparing variants that were detected by sequencing to what would have been detected if a genotyping panel had been performed instead. And what we found is that about 28% of participants, thinking in this case of mostly patients of European descent, had at least one variant, a variant of uncertain significance, or something that's known to be clinically actionable that would have been missed by typical genotyping testing that would have been done in 2022. And notably, if you're looking at, well, what are the genes that are the biggest culprits where the most participants would have had these variants that would have gone undetected? Well, CYP2D6, which is definitely known in the pharmacogenomics community as a challenging gene, but look at that, DPYD, 630 participants, that's 6% of patients, had variants that would not have been detected by a panel at that point in time. So then you think, well, if it's only a couple of variants, but a lot of people have them, maybe you could just add them to your genotyping. But it turns out if you look at DPYD, it was 112 unique variants. So you can't really add all of those to your genotyping test. And for CYP2D6, similar 103 unique variants. So this is something that we've really been thinking about because, especially at Mayo Clinic, we've been doing DPYD sequencing since we went live with the DPYD genetic test, and we also offer genotyping. And so we keep thinking, well, should we continue doing genotyping or should we at some point just switch to only sequencing for DPYD as well as for the rest of our pharmacogenes? But today we offer both for this gene. And one of the things that is interesting to think about is there's these standards and guidelines that are used for diagnostic testing. So this is by ACMG and AMP that had come out with these in 2015. But if you think about DPD deficiency, that rare inherited disorder, well, you could potentially interpret those variants using this guideline. But the decrease in enzyme activity is what's important both for that disease phenotype as well as for the increased risk of fluoropyrimidine toxicity. So what we've actually been doing since we started sequencing is using these criteria, but with a few modifications for pharmacogenomics. And so reporting sequencing really does take a lot of thought, expertise, and professional judgment, and an entire team. So we're really lucky to have genetic counselors to work with. And as a team, we classify these variants when we encounter them for sequencing. So just a little bit more on that. This particular system for diagnostic testing, it's really looking at variants on a spectrum from benign to pathogenic. And so as we encounter these rare variants, you're going to have to classify them and figure out where they fit on this continuum. So specific criteria end up being applied as well as the professional judgment. And potentially, especially if you see these variants of uncertain significance, you may be able to reclassify them over time as more evidence becomes available. So the system's fantastic for diagnostic testing, but there's some challenges because technically it was not intended for the interpretation of pharmacogenomic variants. And then also keep in mind that variants that are associated with non-Mendelian complex disorders, low penetrance, so like our DPD deficiency, that's also another one where these criteria aren't quite perfect for some of those scenarios. And so pathogenic and benign, they're probably not quite the right words for pharmacogenomics, and some of the criteria probably aren't quite applicable for pharmacogenomics, but still, we're talking about genetic testing, and a lot of aspects of the framework could still be helpful. And so, what does this look like? So, there's a number of different criteria that you end up looking at when you're classifying a variant. And so, some of them we did have to think about a bit more. So, minor allele frequency, we have to be careful with pharmacogenomics because some of these variants are really common. We really have to be aware of the enzyme structure, critical domains, and amino acids, but that's no different than doing our diagnostic testing. Functional studies are really important, but when we're thinking about pharmacogenomics specifically, you need to also be thinking about pharmacology a bit more in the therapeutic index. And then segregation with disease, probably not so applicable in pharmacogenomics. I guess unless an entire family is getting the same medication, maybe you can find some of those studies, but not very many of them out there. But the other thing to keep in mind is that this particular classification scheme, it's really just saying, well, how likely is it that this variant is causing disease? Or if we try to apply it to pharmacogenomics, how likely is it that this variant is important for that drug response phenotype? But it doesn't actually talk about the degree of change. And so for pharmacogenomics, if we're going to be predicting where somebody falls on that metabolism spectrum, we really need to think about, well, is this variant going to result in no enzyme activity or only a partial reduction? So we have to assign both an allele function and an activity score. And so we actually went back and looked at our sequencing data from November of 2015 to the end of 2024 for DPYD. And what we were looking at is any variant that's classified as a VUS, likely pathogenic or pathogenic. We reported those out since 2015. The early reports didn't include a phenotype like poor metabolizer, intermediate metabolizer. We used a toxicity risk instead, versus the current reports that include phenotype and activity score. So we reviewed all of that data. And what we found is that about 10% of our cases were positive. It was less than 3% of them that actually had a VUS reported, and the bulk of the cases are still negative, 87%. So, how does this actually compare to doing these genotyping based approaches? Well, if you look at the CPIC guideline, they really highlight four variants that are particularly interesting, and those have been adopted for a lot of the research studies that are out there and a lot of clinical tests. And if you only tested those four variants, you're actually going to detect many of these patients who have DPYD variants. So about 8.67% of the overall results will be positive. If you only include AMP tier 1, so that's a few additional variants beyond these four CPIC variants, you're going to identify that 9.3% of patients will not have a negative result. If you include both AMP tier 1 and tier 2, you get a little bit more of a bump up to 9.44%. And then if you actually jump over to sequencing instead, but you say, well, we don't want to deal with those variants of uncertain significance. We're only going to look at the variants that by the ACMG modified criteria are pathogenic and likely pathogenic, you're going to identify 10.4% of these patients as having a positive result. And once you include the VUSs, it goes up to 12.79%. So in general, you do pretty well if you only look at those four CPIC variants, or especially if you're looking at AMP tier 1 here. But remember, what if you're one of those patients that doesn't have one of these variants but has something rare that could have been detected by sequencing? I think those people would argue that maybe sequencing is a better approach. So, some of the challenges of sequencing, as I mentioned before, if you're doing germline diagnostic testing, the clinical laboratory is going to use these systems to independently classify the variant as they detect it. In pharmacogenomics, the paradigm has historically been different. The variants are typically curated by expert panels that CPIC coordinates, and they assign a function and a strength of evidence. And so, again, the big difference between the ACMG and AMP system from what CPIC has been doing historically is not only who's doing the classification, but also the AMP ACMG system is looking at the likelihood of clinical significance, but not accounting for effect size. And that's something that the CPIC panel does account for, and that's really important. So if we look again further at our retrospective sequencing data, if we're just looking at those benign, likely benign variants or the normal function variants, we didn't track them on a case by case basis, but we know how many total of these variants we've identified. And so, of our unique variants that we've identified, if we look at the databases that CPIC has curated by their expert panel, only 17% of the benign and likely benign unique variants that we identified are actually included there. So this would be a problem for us clinically if we had to rely on just the expert panel, because we would see many other variants that ultimately we classified as things that would not impact DPD activity, but we wouldn't know what to do with them clinically if we didn't have a way to classify them ourselves. And similarly, if we're looking at the other end of the spectrum, looking at unique reportable variants, you can see the breakdown of what we've seen in our lab. Most of them, or many of them, do end up being variants of uncertain significance. Again, looking at unique variants, not number of patients. And the likely pathogenic and pathogenic variants, though, there are definitely some of them that we've classified. And if you look at which ones have already been classified by the expert panel, for the pathogenic and likely pathogenic, it's only 12% of the variants versus the rest of them. Without a system on our own, we wouldn't know how to classify those. Versus the VUSs, again, it's only about 16% of them that were not classified by, or that were classified rather, by the expert panel. So if you're looking at clinical diagnostic testing, there's another group, there's ClinVar, that laboratories will submit variants to with their classifications. And many of these DPYD variants are actually present in the ClinVar databases as well. But again, of the pathogenics and likely pathogenics, 65% of them were in the ClinVar database versus only 12% being classified by CPIC so far. But there's still 20% of them that weren't included in the ClinVar database that we needed to be able to independently classify. And then kind of similar story on the VUS side, about 35% of those were already in ClinVar versus over half of them that we would need to classify on our own. So what I'm trying to really get across as a message here is that at the end of the day, we think the sequencing of DPYD is really important to detect those rare variants, probably similar for across pharmacogenes beyond DPYD. But if laboratories are going to be able to do this themselves, we really need to be able to have systems in place so that we can have consistent classifications. So I think that's just something to be thinking about as a field, because we can't always rely on expert panels if we want to be able to sequence in real time. So some of these challenges, I think it's time really for pharmacogenomics to start creating a framework for variant interpretation. And again, the expert panels, they just can't pre-classify everything, but the expert groups will still very much have a role in this space. And so this is something that as a field, the CLIN PGX group is starting to propose. And so I think we'll see more changes in this area coming forward. So these VUSs, though, I think they're still going to remain a bit of a problem for us. So again, if you're doing diagnostic testing, we consider those to be not actionable. For pharmacogenomics, it's a little harder to know what to do with some of these variants, because some of the variants that we call the VUS may have already been classified by the expert panel, but as having limited evidence to support their classification. And so you really have to figure out, well, how are you going to handle these if you see them clinically? And some groups choose to start with a reduced dose and monitor carefully. Others don't adjust the dose, but still monitor carefully. So this is an area that the field could work on. And so I think it's really good news, though, that less than 3% of our reports actually had a VUS. But what else can we do to decrease the number of VUSs? Well, here's just a screenshot from a study that had been performed way back in 2014 by Dr. Offer et al. And they were doing functional studies in vitro to evaluate the effects of genetic variants on DPD activity. And this is just one piece of data that can help us to work towards classification of our variants. But again, for very, very rare variants, it's still going to be really difficult to be able to get to a definitive classification. So the functional studies have historically been very labor intensive and time consuming, but something that's really exciting is that there are newer methods that are multiplexed that can test many variants simultaneously. There's computational and in silico tools that have always been used in variant classification, but we've not considered them reliable alone. But with new AI tools, this may get better and better over time. And are there ways to incorporate functional studies to even better train the AI tools? Time will tell. So if we move from Sanger sequencing that we do today to next generation sequencing, this is something that's going to be coming in the next few months here. One thing I wanted to mention is that it's going to give us an opportunity to detect copy number variants in DPYD, which we haven't been able to do in our laboratory up until this point. And so, how important is copy number variation? Well, just looking at the NOMAD database, which is a very large population database, you can see that some of these deletions can be up to 0.77% in some populations. And so, thinking about the frequency of some of our tier one and tier two variants, that's pretty important. So, I think it'll be interesting to see how many copy number variants we end up detecting. One last thing to mention, something that's maybe a potential game changer. There was this antidote that came out not that long ago, uridine triacetate, for fluoropyrimidine toxicity. I guess it was approved in 2015 now. And for patients that were in the trial, the mortality was significantly lower than what they had seen before among people who had developed DPD tox or fluoropyrimidine toxicity. So I thought that this was really exciting. Maybe you wouldn't have to always do testing if there's a readily available antidote. However, it's still pretty expensive, not widely available, and you have to be able to recognize the toxicity early to be able to administer it promptly. And so I think it's unfortunately not changed it up as much as I had hoped. And so, really, I think sequencing and genotyping are still going to be really important ways to hopefully prevent some of these toxicities. So, again, why is everyone talking about DPYD? Well, many laboratories are currently implementing this testing for this gene because the demand is very quickly growing. Pharmacogenomics is expanding at this point beyond the labs that have just always done it, in part due to this particular gene. And I think we're going to see the field moving towards sequencing in the coming years so that we can really do a better job of detecting those rare variants and also making sure that our tests are inclusive. But to get there, we're going to need this framework for variant classification. So the real question, should we be universally testing everyone who's going to receive a fluoropyrimidine? Well, not that long ago, all I could say was that the preemptive testing was recommended by the European Medicines Agency. But now, since October 2025, the FDA is also recommending this approach. Tests are accessible, costs have decreased. The pharmacogenomic based dosing has been shown to not negatively impact efficacy, and the guidelines support testing. So I think we're pretty much there. So everyone wants the safest, most effective treatment possible for each individual patient. And so I think one of the things that can get us there, at least for fluoropyrimidines, is working towards incorporating DPD testing more broadly. So with that, I will stop and take any questions.
Thanks so much, Dr. Moyer. And as Dr. Moyer mentioned, at this time, we'll go ahead and answer any questions that you, our audience members, have submitted via the Q&A feature in Zoom. And actually, while we're giving folks a moment to type in any questions that they'd like, and sometimes that can take a little while, Dr. Moyer, I'm going to go ahead and ask one of the questions that was pre-submitted prior to our session today, if that's okay. And that question was, what is Mayo's plan for adding the Association for Molecular Pathology, or AMP for short, Tier 1 and Tier 2 DPYD variants to its tests?
That is a fantastic question. So today, by sequencing, we can detect all of those variants and more. So we really recommend using the sequencing-based test. But for our genotyping test, we've always tried to be very inclusive in our allele selection. But there are a couple of additional alleles that we would like to add, and we are actively working on that right now. So stay tuned to see some updates to both the sequencing and the genotyping test coming soon.
Thank you. Next question is, now that the FDA and NCCN both recommend pre-screening, what kind of growth have you seen in your test volumes?
Oh, that's a great question. So the reason I titled this talk, why is everyone talking about DPYD? Literally everybody is talking about DPYD. I'm getting questions from colleagues who are at laboratories across the country who've never done pharmacogenomics before that are working on implementing this testing. And certainly our test volumes have just grown very, very rapidly in this space. So I think the word's out there, and I think that's really exciting. And I hope we do see a big difference for our patients by implementing this test. I'm just really excited to see this change in the field.
Thank you. Next question is, how much does the DPYD test cost increase when changing from genotyping to NGS?
Oh, that's a really difficult question. And so I have a lot of colleagues who work directly for Mayo Clinic laboratories. And so if you're interested in implementing this testing, or if you're already using genotyping and you'd like to switch to sequencing, I'd recommend talking to the the representatives that work with your laboratory, and they can help figure out what that pricing would look like for your specific institution. And if you're not sure who that person is, you can definitely reach out after the webinar and we can get you connected with the right people. But I'm hoping that the price difference won't be substantially different, such that the sequencing is something that's accessible and available. Because if you can do sequencing, I would definitely recommend going that direction over genotyping.
Thank you. Next question is, have you seen insurance cover all level of testing for tier 1 and tier 2 variants and sequencing?
That's kind of an interesting question. So I'm a little bit sheltered in the laboratory from some of how the insurance coverage works. But one thing I can say is that I was really excited to see that the Medicare contractors are covering DPYD testing at this point in time. And I'm not sure that there's really a difference between sequencing and genotyping, but every single insurer has their own rules and regulations. So I wouldn't want to speak too broadly, but you'd probably want to look into an individual patient's insurance to figure out what it will or won't cover if the cost is going to be a significant concern for them. But I really hope that with more of these recommendations being in place, including the FDA label and the NCCN guidelines, I really hope that means that a lot of the insurers will be covering this testing going forward.
Thank you for that. And then final set of questions from the wonderful Dr. Kolilot. Hi, Dr. Kolilot, out at OHSU. In your experience, are only oncologists ordering these tests? I plan to bring this testing up at OHSU, yes, and wondering who are the other stakeholders? And then following that, does this testing require prior authorization?
Those are fantastic questions too. So I would say that for the most part, the oncologists are going to be the people that will be prescribing fluoropyrimidines. And so I think they probably are the main stakeholder group that would be ordering this testing. However, every institution does this a little bit differently. And especially knowing that this gene does have the inherited disorder that's associated as well, some institutions will do more robust genetic counseling up front. Some institutions may even incorporate in clinical genomics into these discussions to help with testing. But then also, sometimes the pharmacists are really helpful in ordering pharmacogenomics as a whole. And so it could be different areas, but I would anticipate that the oncologists are going to be the primary group that are going to be ordering the testing. Um, Now I am trying to remember what the other questions were. I was excited about the OHSU bringing up the testing.
Me as well. Yeah, it was just a prior authorization question. And
Oh, the
again,
prior authorization.
yeah.
Yeah, I think it really depends on your particular institution and how you handle things. At our institution, this test does not go through prior authorization, but I think it may depend on a patient's insurance and how your institution handles things. So I'll defer to your group to think through it and happy to chat more offline if that's helpful.
Thank you for that. And questions keep trickling in. So in the interest of time, I'm going to make this the last one. Thank you so much for your engagement, audience members. And thank you again, Dr. Moyer. This last question is, what about biochemical monitoring of fluoropyrimidine metabolites in serum as recommended in France? Can it help to understand VUS impact?
That is a fantastic question, and I almost put it into this talk. So it's really interesting because the biochemical testing, it does get used a bit more in Europe than it does in the US at this point. I'm not aware of any labs that offer it right now in the US outside of the congenital disorder and diagnostic testing related to that. But I do think it's a promising way to go about doing some of this testing. If you think about the thiopurines and TPMT, we have biochemical tests for TPMT activity, so it doesn't seem like too big of a stretch. From what I understand, you have to be a little bit careful about test design and specimen handling for these tests and make sure that you've got the sensitivity thresholds such that you can detect the people who would be heterozygous, since they may also be at increased risk of toxicity. But it's something that I think a lot of people are interested in the US. And personally, as someone who does genetic testing, I'm especially interested because I would love to use that as another way to help us resolve some of the VUSs. So I think that's an emerging area. And I think if you keep watching the literature, I think that's something we're going to hear more about in the coming years. It's a great question.
Agree. Thank you so much. And thank you, everyone, for participating in today's webinar. If your questions were not addressed during today's session, please include them in the evaluation, and you can find a link for this evaluation in the chat. As a reminder, laboratory credit is extended for this program, and so upon completion of your evaluation, you'll have the option to claim credit and print your certificate. And final reminder, this program was recorded and will be archived and posted to our website in a few weeks. Thanks again for joining us for today's webinar, and have a great day.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.